The overall goal of this procedure is to analyze the expression of surface and intracellular antigens on neural cells using flow cytometry. This is accomplished by first harvesting neural cells from either in vitro cultures or primary neural tissue. Next, an optional non-specific intracellular stain is applied to the cells. Then, the desired surface antigens are immediately labeled with a primary antibody, whereas the desired intracellular antigens are labeled after cellular fixation and permeabilization. Ultimately, results can be obtained by using flow cytometric analysis for co-expression of the surface antigens on specific neural subpopulations of interest, as defined by intracellular label. This protocol will demonstrate how to prepare tissue culture-derived neural cells for interrogation of surface and intracellular antigens with multicolored flow cytometry. To begin, harvest adherent neural cells grown in T75 flasks by first gently washing the cells once with 5 milliliters of magnesium and calcium-free PBS at room temperature. Then, detach the cells from the flask by adding 3 milliliters of trypsin replacement and incubate at 37 degrees Celsius for 2 to 5 minutes. Quench the enzymatic digestion by adding 6 milliliters of flow buffer. After loosening the cell clumps by pipetting the cell suspension up and down a few times, transfer the entire